Hello everyone, we are from Aspergillus group. We'll present about the concept of D and Z value in controlling growth of microbial process by using sterilization and pasteurization method. Okay, first we are using canned fish product as our food type. For example, tuna, smoked fish and salted fish. So this kind of food product are using sterilization as processing method. So sterilization is a process which destroys microorganisms including bacteria, sporogenous forms and inactivates virus. The destruction of microbial population follows a logarithmic projection which is by D and Z value. So D value denotes decimal reduction time. It is the time required at a specific temperature and under specific conditions to reduce a microbial population by 1 decimal or to kill 90% of microorganisms. So in simple words, D value is time needed at certain temperature to kill an organism. The decimal reduction time is depend on the temperature, the type of microorganism and the composition of the medium containing microorganism. Next, about Z value. So Z value is the increase or decrease in temperature required to reduce or increase the decimal reduction time by 1 decimal. It is also a measure of the change in death rate with a change in temperature. So do you ever think what kind of bacteria that usually found in canned food? So the bacteria that usually found in canned food is Clostridium botulinum. The endospores of this bacteria is the causative agent of botulism in canned food. So this bacteria is a gram-positive, have rod shape, anaerobic and spore forming. So this bacteria also have ability to produce the neurotoxin botulinum. The D value of Clostridium botulinum is reduced by 90% after exposure to temperature of 215 Fahrenheit for about 0.1 until 0.23 minutes. Meanwhile, for Z value for this bacteria, every 10 degrees Celsius change in temperature, there is a tenfold change in its death rate. Next, the growth factors of Clostridium botulinum. So first is the environment factor. This bacteria is an anaerobic bacterium which can only grow in the absence of oxygen. Next, the level of acidity. So the higher the pH, the greater the level of heat needed. Next, the water activity for this bacteria is about 0.94 until 0.97. And finally, the temperature. So the optimum growth temperature of this bacteria is about 26 until 13 degrees Celsius. So for the next food type is we are choosing dairy and non-dairy milk products. The processing method for this food product is by using pasteurization method. Pasteurization is a process in food such as milk and fruit juice. It is treated with mild heat usually to less than 100 degrees Celsius to eliminate pathogen and extend shelf life. The process is intended to destroy or deactivate organisms and enzymes that contribute to spoilage or risk of disease, including vegetative bacteria, but not bacterial spores. So let's see about D value in pasteurization. So the heat process is usually based on 12D concept or 12 log cycle reduction in the number of these organisms. For example, a D value at 72 degrees Celsius of 1 minute means that for each minute, bacteria population of the target microorganism will be reduced by 19%. Next, the Z value in general form. The Z value reflects the temperature dependence on the reaction. It is defined as the temperature change required to change the D value by a factor of 10. The reaction that have small Z value are highly temperature dependent, whereas those with the large Z value require larger change in temperature to reduce the time. Meanwhile, Z value for milk, due to the difference in Z value, it is apparent that at higher temperature for shorter times, a region exists where pathogens can be destroyed while vitamins can be maintained. For example, in UH3 meat processing, very high temperature for very short time are favored compared to a lower temperature with longer time process. So how to calculate the D and Z value in pasteurization? So let's see the example here. For example, full raw milk at the processing plant has bacteria population of 4 times 10 xp 5 per milliliter. It is to be processed at 79 degrees Celsius for 21 seconds. The average D value at 65 degrees Celsius for the mixed population is 7 minutes, while the Z value is at 7 degrees Celsius. The D value has been reduced by 2 log cycles at 65 degrees Celsius since the Z value is 7 degrees Celsius. Hence, it is now 0.07 minutes. The milk is processed for 21 divided by 16 equal to 0.35 minutes. So that will accomplish 5 log cycle reduction to 4 organisms per milliliter. So at 65 degrees Celsius, you will need 35 minutes to accomplish a 5D reduction. Lactic acid bacteria commonly found in dairy products, for example, Streptococcus sp. So the growth factor of this bacteria is often not manufactured under sterile condition or with sterile meat, and this can allow the pathogenic bacteria access to the fermenting food system. The growth factors of bacteria that commonly found in dairy product is pH, water activity, and salt in moisture levels as well as temperature. The optimal pH for the thermophilic lactic acid bacteria, for example, Astemphilus, was between 6.5 and 7.5. For water activity, the growth of most bacteria in milk is in range between 0.90 until 0.91. Finally, for the temperature, bacteria associated with dairy fermentation can grow over a wide temperature range from 4 to 15 degrees Celsius. So that's all from us. Don't forget to like and share this video for educational purpose. Thank you for watching this video and have a nice day.